Students, here I am discussing the topic molecular basis of inheritance, which belongs to the 12th standard, chapter number 6. This is very important topic which deals with the inheritance pattern, which is basis on the molecules. We have to study the genetic material as at molecular basis. Here I am discussing the replication of DNA, particularly in the eukaryote, not in the prokaryote. Generally, student, there is a question when the replication of DNA is start and where it occur. Particularly, the replication of DNA start during the cell division, before the cell undergone into the division, during the interphase. Interphase having the three events G1, G2 and S phase. So S stands for the synthesis. Here in the S phase, the replication of DNA is occur or synthesis of DNA is occur. Here I am discussing how the replication is occur. So for this purpose, I am choosing this representative diagram which indicates the two strands of the DNA, because DNA have the bipolarity nature. There are two different types of ending of the DNA. One is 3' and its another ending ended with the 5'. 3' indicates the hydroxy group and 5' indicates the phosphate group to its end. Okay, so let us discuss how the replication of DNA is occur. Replication process require some requirements. These are as deoxyribonucleotides. Deoxyribonucleotides, friend, are previously present in the nucleoplasm of the nucleus. Nucleoplasm is the fluid of the nucleus. So, inside the nucleoplasm, deoxyribonucleotides are present. Activation of the deoxyribonucleotides occurs with the help of ATP enzyme. Another enzyme which is known as chain polymerase. In the eukaryotes, there are 5 types of chain polymerase enzymes, that is the alpha, beta, gamma, delta and epsilon. There are 5 different types of chain polymerase enzyme. While in the prokaryotes, there are only 3 polymerase enzyme: polymerase first, polymerase second and polymerase third. Here, the alpha, beta, gamma, delta and the epsilon, 5 different type of polymerase enzyme are required. Here the alpha, delta, epsilon are the major polymerase enzyme, while the beta polymerase enzyme act as a proofreader, or it removes the wrong base pair during the replication. Some mistakes are occur. So when the wrongly base pair is added to the newly strand, so this beta polymerase chain enzyme remove that wrong base pair. So mistakes should be corrected by this proofreader, that is known as beta polymerase enzyme. Next enzyme is helicase, which is also known as unbinding protein, which unbinds the DNA by means of this. This is the unbinding of the DNA by means of the helicase enzyme. Helicase that breaks the helix of the DNA. Here is the helicase enzyme. One more enzyme which is known as endonuclease, which cuts the DNA strand by means of breakdown of hydrogen bonds to its inner side. That's why it is known as endonuclease enzyme. The enzyme pyrophosphatase. Pyrophosphatase is helpful to remove the extra phosphate group from the deoxyribonucleotides, and which makes the nucleotide with the single phosphate group. Because we know that after activation, the addition of 2 more phosphate group by means of this ATP reaction, this get removed by the pyrophosphatase enzyme. The next enzyme is the ligase enzyme, which seal off, which act as a biological glue which seal off this lagging strands. And the last one is the topoisomerase enzyme. Topoisomerase enzyme which is present at the end of this binding site of the enzyme. Okay. This topoisomerase enzyme save the DNA from the breakage. Okay. So this is all about the accessories or the requirement of the process. This is the systematic diagram. During the replication, the DNA is get unwind with the help of helicase enzyme, and the activation of deoxyribonucleotides occur in the nucleoplasm of the nucleus with the help of ATP enzyme. One more requirement is RNA primer. RNA primer are the short sequence of RNA molecule. These short sequence of RNA primer are also present in the nucleoplasm of the nucleus. This RNA primer either gets synthesized by the nucleolus of the nucleus, or it is synthesized at one of the end of DNA template. It is attached to the 3' end of the template. So once it added the nucleotide one by one from the nucleoplasm with the help of this polymerase enzyme, it moved forwarded. So we can say that the RNA primer acts as a starter. Please note it. In the eukaryotic cell, there are certain or many origin site where the DNAs get cutted, or the nicking site. Nicking, that is the incision site where the DNA is cutted and start to unwind by means of this helicase enzyme. Now each strand which get relaxed due to the activity of this helicase enzyme, now there is a possibility to rewind or recoil once again. So this recoiling gets stopped by the enzyme single stranded binding protein, which makes each strand remain free and not undergone into the recoiling. Now the student, to this RNA primer, the DNA polymerase is get attached. The replication of DNA, particularly in the eukaryote, is studied by the scientists Messerson and Stell in 1958. According to them, the replication is semi-conservative mode and it is semi-discontinuous process, which is always takes place in the direction 5' to 3'. It never comes from the 3' to 5'. That means in the eukaryotes, the replication always unidirectional. Such points are asked in the competitive exam because there is a difference in the prokaryotes. The replication is bidirectional, while in eukaryote the replication always unidirectional manner. It is takes place 5' to 3' direction. So once the RNA primer gets synthesized at 3' end, both the strand of DNA act as a template. Template means साचा, जिसमें कोई copy की जाए. So both the strand act as a template. So the RNA primer once gets synthesized at the 3' template, 3' end, the DNA polymerase is attached to it and start up the process. What I said to you, the DNA replication in eukaryotes takes place in 5' to 3' direction. So it is started from this to the 3rd' template. The RNA primer start up the process in the manner 5' to 3' with the help of DNA polymerase enzyme. The activated deoxyribonucleotide added one by one. So one strand which was formed on the DNA is continuous and another strand which was formed during the replication in a discontinuous manner. Why so? Because the unwinding process requires the time. That's why the another strand is formed in a discontinuous manner. Whatever may be so, but the replication of eukaryote always takes place in a unidirectional manner. Here, this strand which is continuously formed is known as leading strand because it is continuously run from 5' to 3' direction, which is the new strand, and the another strand which is formed slowly by the slowly opening of the strand at the top, so it will form in a discontinuous manner. It is termed as lagging strands. Here also, this is the model which is the inverted Y-shaped model which is studied by the scientist, Japanese scientist Yabuta. Yabuta said that this Y-shaped model indicates the replication manner of DNA. This cutted discontinuous newly formed DNA in the pieces said that Oki-Joki fragment. Oki-Joki fragments are nothing but the lagging strands or the discontinuous replication of the DNA, while this continuous newly formed strand is known as leading strands. So in this manner, with the help of this sort of enzyme, the replication get completed. Here the student, I am using the blue marker for showing the old strand and the red colored marker for showing the newly formed strand. So in this manner, the two strands were old and two are new one. That's why Mesoscale said that this replication mode is semi-conservative. 50% of DNA is of parental type and 50% of DNA is of new type. This diagram indicates. Now here I am not showing the bonding between the base pair because before the proofreading, bonding not takes place. Once the ligase enzyme stick the or seal this lagging strand, which was the discontinuous one, the gap inside this discontinuous strand get filled up by the enzyme polymerase, and this ligase enzyme, which is act as a biological glue, seal this lagging strands and makes it continuous. Now this old and new strand undergone into the bonding. Before the bonding or base pairing occurs, the proofreading is must. During the process, during the replication process, so many mistakes are occurred because this chain polymerase enzyme cannot recognize the difference between the uracil and the thymidine or the thiamine group, thiamine base pair, because there is a similarities between the structural similarities between the uracil and the thiamine. So sometime what happened, this alpha polymerase added wrong base pair during the replication. So meanwhile the beta polymerase and the topoisomerase act as a proofreader. They remove the wrong base pair from this replicate fork, and then only the hydrogen bonding occurs in this. Okay student, once the hydrogen bonding is occur, these two strands which was running anti-parallel to each other now get twisted together, and the RNA primer and the DNA polymerase get removed. So in this manner, the replication of DNA is occurs successfully without the mistakes. Okay, thank you student. So here I